Welcome to the deep dive. Ready to explore how gadolinium contrast can change the game for susceptibility-weighted imaging in canine brains?
Absolutely. It's a niche topic, but the findings are pretty significant for how we use SWI in veterinary neurology.
SWI is already an amazing tool, right? It's so sensitive to magnetic susceptibility differences.
Yeah, meaning we can see really subtle changes in tissues.
And visualize things like blood and iron deposits in the brain.
It's like getting a microscopic view of the brain's vasculature and iron distribution, which is so important for diagnosing a whole bunch of neurological problems.
This deep dive comes from a 2024 research article out of Veterinary Radiology and Ultrasound. They studied 71 dogs looking at one key question.
Does using gadolinium, that common MRI contrast agent, mess up SWI results?
And to figure that out, they split the dogs into two groups.
Right, so Group A got gadolinium and then had an SWI scan immediately after.
Gotcha.
While Group B also got the gadolinium, but their SWI scan was delayed by about 20 minutes.
So the timing of the scan was the variable?
Exactly. They wanted to see if that affected the SWI results.
What do they find?
Gadolinium actually caused an increase in signal intensity in certain brain areas, especially in the white matter.
Making the signal brighter after giving the gadolinium.
Precisely. They look specifically at a region of white matter called the centrum semiovale, and this increased signal intensity was most noticeable in Group A, the dogs that had the SWI right after the contrast.
So timing did play a role.
Yes, it did. But this increase in signal intensity wasn't seen in the gray matter, specifically in a region called the lentiform nucleus.
That's a big difference between white and gray matter. Why might that be?
Well, one possibility is that the composition of white and gray matter in dogs is different than in humans.
Interesting.
The researchers think this difference could affect how gadolinium spreads in the brain, leading to those varied signal intensity changes.
Makes sense.
Yeah.
Different structures, different responses. To measure these signal intensity changes, they used what's called ROI analysis. What is that exactly?
ROI stands for region of interest. They basically drew little virtual boxes around certain areas on the SWI images, like the centrum semiovale and the lentiform nucleus.
Okay.
So they could precisely measure and compare the signal intensity within those defined regions before and after giving the gadolinium.
A way to zero in on specific areas and track the signal changes.
Yes, and here's where it gets even more interesting. This finding in dogs is actually the opposite of what's been seen in human studies.
Really?
In humans, gadolinium hasn't seemed to impact signal intensity in either white or gray matter.
So there's a species difference there. The study also looked at how gadolinium affects brain lesions. What did they find there?
Gadolinium made the signal intensity go up in the brain lesions too, and this happened in both groups no matter when the SWI was done.
So the lesions got brighter after the contrast, whether the scan was right away or delayed?
Exactly. And this could be a really big deal for diagnosing stuff in animals. Think about it. If we can consistently see this signal change in lesions, it might help us tell the difference between different types of lesions.
Like, we could maybe tell the difference between inflammation and a tumor based on how it reacts to gadolinium.
That's the idea. It could change how we diagnose and treat neurological problems in dogs. It's similar to how they use contrast enhancement to look at brain tumors in people.
Wow, that's really exciting. Every study has limitations though, right? What were some of the limits in this study?
Well, the sample size was one thing. 71 dogs is okay, but having more dogs would make the findings stronger.
Makes sense. More data is always better. What else?
Another thing was that only one person looked at all the MRI images. It would be better to have a few different experts look at the picture separately to make sure there's no bias.
Yeah, that makes sense. Anything else?
They also decided to just look at the biggest lesion when there were multiple lesions, so they didn't really look at every single lesion, which could have meant missing some details.
Right, so they made the analysis easier, but maybe lost some information along the way.
And lastly, the diagnoses were made using what they saw clinically, what the MRI showed, and cerebrospinal fluid analysis, but they didn't confirm with histopathology, which is the gold standard.
Right. But it's not always possible or ethical to do histopathology for every case. So even with these limits, the findings are still pretty important.
Definitely. This study opens up some really cool possibilities for the future of neuroimaging in animals.
So how do you think these findings will affect how veterinary neurologists and radiologists work in practice?
Well, first of all, it shows that gadolinium doesn't mess up how we interpret SWI images, even though it can change the signal intensity in some places.
That's good to know. We can keep using gadolinium without worrying.
Exactly. And secondly, it suggests that these signal changes, especially in lesions, could become a really useful tool for figuring out and maybe even grading different brain problems in dogs.
So it's not just about seeing the lesion, but about understanding what it means.
Precisely. And that could have a big impact on how we treat and manage neurological conditions.
This feels like a big change in veterinary neuroimaging. It's like we've got a whole new way of looking at the brain.
It's a powerful tool, and I think it'll get even more important over time.
Of course, more research is needed to confirm these early findings and explore all the ways we can use them.
For sure. We need bigger studies with more people looking at the data and ideally histopathology confirmation to really back up these findings and build on them.
Absolutely. But this study is a great first step. It shows how much progress is being made in veterinary neuroimaging and how it can help us take better care of animals.
Speaking of which, the researchers ended their article with a really interesting question. Want to hear it?
Definitely, I'm all ears. I'm all ears. Tell me.
They asked, could this difference in signal intensity after we give gadolinium be used to better characterize brain lesions in dogs?
Now, that's the real question, right? The study seems to show a difference, but can we actually use that for something?
That's the crux of the matter, and that's what we need to research more. If we can really tie specific signal changes to certain types of lesions, it could totally change how we diagnose and treat things in veterinary neurology.
It would be like having a map of the brain, right? Not only seeing that there's a lesion, but knowing what it's made of and how it's likely to behave.
Exactly. Imagine being able to confidently say, okay, this lesion looks like inflammation, or this one has the signs of a tumor.
Yeah, that would be amazing.
Yeah.
We could intervene much earlier and more precisely, which could mean our patients do better.
Absolutely. That's why this study is so important. It's not just about what they found, but about all the possibilities it opens up for future research.
It's like they gave us a key, and now we have to find the right lock.
Exactly.
Yeah.
And I'm sure veterinary neurologists and radiologists will figure it out. This field is always pushing the limits, and I can't wait to see what happens next.
Me too. This is a really exciting time to be in veterinary neurology. With all the advances in imaging like SWI, we're learning so much more about the brain.
And that knowledge helps us take better care of our patients. That's what really matters.
Well said. Okay, it's time to wrap up our deep dive into gadolinium and how it affects SWI in dog brains.
We've covered a lot today, from how the study was done to what they found and what it might mean for veterinary medicine.
It's been a journey into the complex world of the brain and the amazing technology helping us understand it better.
As always, we encourage you, our listener, to stay curious, keep learning, and keep asking those questions.
Because that's how we make new discoveries and move veterinary neurology forward. From Vet NeuroJar, keep those minds-inspired hearts light and tails wagging.